morning, everybody. Can you online experts here? Can you do a thumbs up, thumbs down for us? Maybe we can only see Angela. Angela, you can see us, right?
Yep.
Wonderful. So welcome everybody to Ontologies, Annotation, Metadata. There might be some other people coming in, but we have a very full program, so we'll see. This is one of some breakout sessions. So as you saw,
you want to hit slideshow?
In that moment I can't take notes anymore, so
Okay, sorry.
Advantage, disadvantage here. Also, this way you kind of preview on the left side. Might be working on it. So it's actually a back and forth between David and myself. David is an ontologist. He works on ontologies. He engineers them. He has been taking very good care of the ontology additions that are coming from HubMap. We believe within HubMap alone, we need about 5,000 new terms to cover the healthy human body. So how do we get those integrated into
difficult to hear.
Okay, I'm very close to this wonderful device. I think that's the one we have. Can you hear us better if I hold this up to my mouth? Is this better? Can somebody use the chat to let us know if this works for online? All right, Angela gives us a thumb up. Wonderful, thank you. Keeps this table here.
I think it's okay. Just put it on the laptop. It should be okay.
So on the other hand, in HubMap, we have a lot of 3D and 2D and also some LinkedIn 1D representations of human organs for male and female. We also have functional tissue units. We of course have many segmented data sets. And all that also has to be captured as part of the reference atlas. So two of us come from very different sides. One is more textual, that's David, and one is more visual, 2D, 3D. Melissa will also present some examples. Irene just presented some of the examples that they did for the anatomograms. And so what we assembled is a set of speakers, which you see here, which come also from these different angles, but also ultimately try to use ontologies to augment and improve machine learning algorithms and also human decision making. But I deeply believe that the atlas cannot be built just using human intelligence alone. We need probably 80%, if not 90% machine learning intelligence. And then maybe would be the human intelligence. All right, so coming over to what you saw on the slides and also on the virtual website, we have agreed on those 4 questions, and that was not easy because again, schools of thought. We decided that there would be flash talks, 1 or 2 per these major questions. And it's wonderful that so many of you agreed to present here just for 5 minutes, but it's important to see your comments on it. And ultimately also have an overview and a welcome from both of you, from both of us. So I think David's turn is first. So he will give his brief welcome, then I give my brief welcome, then we go over to the first out of 4 questions, have a speaker, a quick brief discussion, next question, brief discussion. And all of you, I think I have been invited to a back channel. If you have not received an email from David, this is your moment to say, here, I want to contribute. And then David somehow magically presents and sends you this link. Good luck.
Okay, so I think sorry. Okay, can you go to the first slide again, just quickly? That will give us the link. Anybody who wants to take notes, to add to the notes that we're taking here. So which one is bigger?
I can put it in the chat.
Yeah, I'll put it in the chat as well. So it's bit.ly/HCA_cell_hont, and that's to the notes stuff if you want to contribute generally to the notes. Oh, we're not sharing, okay. All right. Okay, general introduction. Very quickly on why we, some background on why we do the ontologies. I mean, sort of the main issue is making data, one of the main issues is making data findable, accessible, interoperable, reusable. If you go back to that first slide, this just shows a quick example of all of the different names given to a cell type in the COVID cell atlas from 81. So, you know, some of these not even related to each other textually, when many people actually in the literature call this term a type 1 pneumocyte, it might be called alveolar type 1, 81, etc. So we need some level of standardization on the annotation so that we can start grouping things easily. And if you, next slide please, if you go, then the ontology structure itself allows us to group content. So in this case, the hepatic site, this cell is Kupffer cell, which you can see in this nice anatomogram from the single cell atlas. We can group it under liver, hepatic sinusoid, tissue resident macrophage cell because of the structure of the ontologies. The structure of these ontologies is useful in grouping data, in making it findable through various queries, and also potentially in integrating data into a more complete atlas. So really, that's just the main intro stuff I was going to cover. Do you want to just go to the next slide? Right. We have 4 discussion topics. The original plan was we hoped we would be able to do at least 2 breakout rooms with some people going out into a separate room here and using a separate Zoom room. That turns out not to be supported. I guess I'm very used to running these things online now. We're like, okay, 4 breakout rooms, and you're done. So the hybrids made that a little more difficult. So instead, we're going to cover these topics after the breakouts, after the flash talks. The flash talks are going to be super fast. They're 5 minutes. The idea is really just to highlight some issues, some solutions to some of the main questions we want to cover here, the main discussion topics. How can we extend and improve ontologies? We might minimize discussion of that because looking at the slides, there's looking at the presentations, there's not too much on that, and I want to reduce that. We have plenty of time for discussion on specific topics. How can we make ontology annotation following standard schemas easy and efficient? That I think is really, you know, the usefulness of the ontology very much depends on getting annotation out there and making it as easy as possible. And I think Evan's work on the CAT platform and our contributions to that, I think that's one of the key ways that we do this. And Angela and Fabian's work on and many others on being able to project ontology annotations. In the case of Angela, actually using ontologies themselves as one of the inputs using machine learning. I think that's a key part of that. How can we enable downstream users to take advantage of the ontology annotations? And I think Fabian's Esfero library is a really nice example of actually integrating this. You can actually start using it in analysis. And I think, you know, again, the usefulness of this stuff really means we have to be able to plug into all the pipelines of how people are analyzing their data. And then finally, how can ontologies contribute along with all of the other infrastructure we're talking about here, machine learning, etc., to actually building these integrated atlases that we want to build. And I think they're one key part of that picture. So those are the main discussion topics. On the first topic, we're also actually hiring ontology experts. So one way extending and improve ontologies is finally getting enough people to come to terms. It's not actual rocket science. Right at the bottom, we're cutting off the bottom of the slides here, but the reminder for speakers here is 5 minutes only, emphasize problem solution. Katy's got a couple of slides, and then we're going to go straight into 5-minute flash talks, and then the rest of the session with the discussion. So, Katy, do you want to?
Cool. Yeah. So some of you might have seen that there are now 17 consortia collaborating together on agreeing on anatomical structure terms, cell type terms, biomarker terms, biomarkers covering genes, proteins, lipids, metabolites, And that's a huge community effort. It's really wonderful and magical to see it all come together. At the same time, there are also other parts of these experts working on these 2D representations, and illustrations of these anatomical structures and cell types. We are also part of another effort, which is SPOKE, which interlinks many, many different ontologies. All of these efforts are free, openly available, lots of code that surrounds these ontologies. And of course, you want to connect those two, right? You want to connect the healthy human being with disease ontologies and other ontologies that can help us understand what happens in spikes, for instance. So my big question for this event here is how can we bidirectionally link text to imagery or 3D or 1D models, and backwards also from the 2D representations, be it segmented cells or functional tissue units or anatomical structures, back to the standard ontological assigned representations.
If you speak about reference, I don't know this term, so I haven't seen it much. This is like a sketch of how an organ looks like, not like a real data set or something. Is that what you're saying?
So this was, so these 3D models come
Yeah.
from the Visible Human Project data. So they are now, this is the National Library of Medicine, scanning an entire human body and entire human male and female. So that was segmented in 3D and made available, and is now used to register tissue blocks, but also to query for tissue blocks that have been registered into the system.
So this is like a generalization of this common coordinate framework type of thing that they use.
Yes, and then you can take a human being like yourself
Yeah.
and map it into it. There are algorithms that can absolutely do that. But you now have one space in which all of this data can live spatially. And then you can also predict based on the space that a certain tissue block occupies, what kind of cell types are most likely going to be in there. And what kind of validated antibody panels you should use to do analysis. And then inspired by the anatomograms, which Irene just showed, but also KPMG's Atlas Explorer, you now have these 2D representations. And Melissa is going to talk more about that line of research. And you can then connect that to the real world data. So you can use these illustrations and actually start using that to train machine learning algorithms. So just opens the next couple of competitions, this time with the protein atlas. So that's my big question for today. So let's see what questions you bring to the table. So we have now flash talks by experts, and I believe Melissa is first. So note that we reorganized the speakers, so you might not be in the sequence that you had last night. Melissa, please. And I can forward the slide.
Thank you for asking me. So I'm Melissa Clarkson from the University of Kentucky. And so talking about representing human anatomy at the scale of organs to tissues and the foundational model of human anatomy project. So my first slide is to remind us that anatomy is complex, like we didn't know this, but just to remind us. We've got not only differences in spatial scale going from gross anatomy to histological anatomy to molecular anatomy, but also organisms develop over time. And so, unlike the rest of you, I don't work on representing cells. I instead work on gross anatomy and histological anatomy.
I'm sorry that it's kind of.
So next slide. All right, so the foundational model of anatomy is a reference ontology for adult canonical anatomy, and it's the project of the Structural Informatics Group at the University of Washington. And this is modeled in web ontology language, otherwise known as OWL. And this is a 20-year project. It represents over 100,000 anatomical structures as classes, and also has over 100 types of relations among classes. So these are not only part hierarchies, things like connectivity. So moving through paths through like the circulatory system, lymphatics, the way muscles are attached to those types of relations as computational accessible knowledge in the ontology. Next slide. Okay, so I was the last graduate student in this group, and I did my PhD dissertation focusing on the FMA. And as I was digging into this, I found a few issues. So for example, there was variations in modeling schemes. So similar anatomy in different parts of the body was modeled differently due to inter-author variation over this 20 years of development. There's also, unfortunately, quite a bit of incomplete content. So there might be There was lymphatics at the head and the breast and a little of the legs, and the rest of the lymphatics was missing. And finally, a problem I found that was frustrating, but also presented an interesting challenge was it was not easy to understand the meaning of some of these classes. So there was a term, and well, people might interpret that anatomy term differently. And this is a problem if we're trying to standardize annotation. So, next slide. Well, I am a new assistant professor at the University of Kentucky, so I have inherited this project, and I realized instead of continuing the FMA as it was, I really needed to make the next generation FMA. So I am calling this the foundational model of human anatomy, just to clarify that humans. And so what my strategy that my team is using, we are going to improve the consistency of modeling. We're going to improve completeness and also clarity. So to give you an example of what we're doing for consistency of modeling, next slide. So let me start with an example of the inconsistency. So again, we're thinking gross anatomy. So we have muscles and bones. So if you were to computationally query how is the human body, the muscles connected to the bones, you would expect it to be pretty consistently in there, but unfortunately not always. So next slide here. So, so to clarify, we've got muscle organs referring to the entire muscle, and the bone organs refers to an entire bone organ because we don't use the term bone and muscle, that could be such. But anyway, so looking in the FMA, how are muscles attached to bones? So if looking at the superclass of tendon, we've got some that say tendon attaches to zone of bone organ, and there's 207 axioms like that. But sometimes it was the zone of muscle organ has insertion zone of bone organ, there's 60 of those. And sometimes the muscle organ has insertion zone of organ, 36 of those. There's another row on here that I can see, but that's okay. My point is these were not consistently modeled due to inter-author variation over the 20 years of development. So this is the problem. So the solution that my group is taking, next slide, is to come up with patterns. And so here's a very simple, simplified one. We're going to general pattern we're using is that a muscle organ has constitutional part tendon, and that tendon attaches to the zone of bone organ, and that zone of bone organ is a regional part of the bone organ, and there might be some other parts in that shape. So very oversimplified example, but that's what we're doing is we're figuring out the patterns, and then we're going to match the content to this. And next slide. So the vision for this is that the FMHA will be a computable representation of human anatomy. And the part that I had to cut out for time is my graphics were also going to have standardized graphics, make a huge graphics libraries, such that instead of just having to interpret the term, there will be a picture for most of these. So it doesn't matter what language, it doesn't matter how you interpret the term. If we can, it's like, oh, the picture, that's the thing I need. That's how I'm going to help all of us in the world who dealing with anatomy and standardization, with the end product being an illustrative ontology. So that is, oh, I saved over time. That's all I have to say.
Discussion, very quick.
Yeah, quick question. So typically an illustrated ontology would need to capture a general case, but there is no general human being, right? We are always unique and very special. So now you have a very general representation of the human body, point to some very specific thing. And ideally, if you register something into that very specific thing, it comes back to the ontology and can link to the right term. So how do we deal with this? Because there's like almost a philosophical problem there.
So the FMHA is going to, well, the FMA models canonical anatomy. So I'm staying within the framework of canonical anatomy, this magical, fictional human body that is canonical.
Okay.
Some of my side projects are phenotypes and malformations, and then throwing development. I think it's, this is the next decade of my life. So this is not going to materialize in the next year or two, but I'm working on the musculoskeletal system right now, hopefully doing a deep dive into kidney this fall. And so over the next decade.
Yeah, we're doing a lot of work actually to take complex ontologies like this and come up with simplified views. So I think there's good opportunities to work together
Yes.
on taking the amazing 20 years of work that have gone into this and providing really simple views. cut down use.
Yes, and hopefully our patterns, because we'll have enormous amounts of detail in what we model, and if you just want to say, no, I just want to see muscles to bones, I don't care about all the parts in between, you can use our patterns and cut it down just to the simple part.
Next up, Wei is going to talk about the DCP.
Hi everyone, I'm Wei King. I'm here to talk about the Human Cell Atlas data portal. I'm from the EBI. So today I'm going to be talking about how we can make ontology annotation following standard schemas easy, efficient, and accurate while leaving room for revising and adding to existing ontologies. I've got two main points. The first point will be about the Human Cell Atlas data coordination platform metadata schema, how we use and validate ontologies there. And the second point will focus on cell ontologies and cell level ontologies and annotation. So super quick overview, the Human Cell Atlas data coordination platform is a platform for researchers to interrogate single cell data. We gather a lot of metadata, we put it to a specific standard, and we are a open, organized, and dynamic platform that tries to grow and change according to the needs of the users, which are you guys in the room and everyone else who is part of the global community. Next slide, please. So the thing that we mostly do at the Human Cell Atlas is that we have a very high standard for metadata that is required because metadata is golden in our eyes. We gather lots and lots of different metadata, known diseases, ethnicity, organ parts, and we want to make that all available for scientists because what is important for one researcher might not be for another, and we try and gather as much of that as possible to have this high quality metadata standard. And so in terms of our metadata schema, it is a JSON format and it contains all the information required to understand and interpret the standard. Here we've got two entities, organism age and organism age unit, and by the JSON schema, it validates against the data and JSON files according to specific rules. So in terms of ontologies, the schema can be expanded to validate for them. We have here an entity for organism age unit, and only a term under the classes time unit or derived time unit will be accepted. Any other term not descending from these two is rejected in this field. So it's just a very clear, straightforward representation of how we can use the schema to validate for the hierarchical nature of ontologies and accept some and restrict it to a specific subset. So as a result of our validation, we also have ontology terms for a lot of metadata. So we have ontology terms for ethnicity, developmental stage, age unit, so on and so forth. And this results in us having a high degree of harmonization between all of the different projects that we have available on the data coordination platform. They all have these ontology terms, and you can filter and search through them very, very easily. So on to cell ontologies and annotation. One other thing that we've been working on in the HCA is sending and wrangling data sets to cell by gene according to their specific schema. What we do is take that list of ontologized terms from the HCA, that large amount of metadata, take those ontology terms at the moment as subsets and link them directly within the expression matrix. So you have a link between your cell level, your cell barcode, and each individual cell there, and you can link that all the way back to the donor, the protocols that were used, the organ, the ethnicity, how many smoking packs that specific person used if that data was available, and we managed to wrangle it into the DCP. So all of that metadata is searchable on cell by gene, and you all know cell by gene. I don't need to talk that much about it, but that is one of the ways in which we take the ontologies and make them useful there. In terms of cell type ontologies, cell by gene has a requirement for the cell types to have ontology terms, and so we often receive cell types from contributors or publications in the form of free text. So we work with them and with the CL team to make sure that the most accurate or most precise ontology term is used to represent the cell type. And obviously this comes on to some of the pain points and challenges in future development. What happens when we receive a free text cell type that doesn't have an ontology term, which happens a lot. At the moment, we're working together with David Osumi-Sutherland's team and the very aptly named CAP that was spoken about earlier, and we're trying to figure out a way for minimal metadata required to turn these free text cell types into ontology terms. And I'd also like to talk about the importance of releasing annotations of data, which Fabian spoke about earlier, and how important that is to have those annotations. And lastly, to talk to us here or email us.
Oh,
Perfect. Quick question. How many new cell types do you add per year?
So the work that we're doing with Cell by Gene is still quite recent. We've only been working with them directly and urgently on this over the last 2 months or so, but we've come up with a bunch of different new cell types. We're piloting one data set now with David, which has 6 different cell types, which don't precisely have ontology.
I think it's, immune cells sort of go on forever. The brain, there's lots of new cell types, but for other tissues, it's a smaller number of new and more specialized types generally.
I think we need to have this hierarchy here. This is quite important. Right?
Yeah.
I think this is kind of supported. I'm wondering how this works. So is Cell by Gene going to be a superset of everything you have in DCP? So everything is going to be forwarded to Cell by Gene portal as well?
At the moment, we're
That's
focusing.
nice.
Jason's going to talk about.
That's the hope,
Cell by Gene.
yeah.
So,
That's the
so
dream.
I think we need to be really clear to the community, where do you guys go and like you check out things that's too many portals and.
And
Yeah.
It's too much effort.
Yeah.
Nobody can do that. Just move it on.
Yeah, right. So I mean, one outcome of this thing is, we draw a dependency graph of what's where and like where can you go. So then we have this file, we just like know from the
Centralizations of
Yeah,
data.
so actually
Yeah.
giving people a flowchart of where to go depending on what their data is and how that.
I think that would be
really useful outcome. Yeah. I
Because
really like that.
Because if you don't tell people where to put their data, they might not put it anywhere.
There might be some argument about.
Well, they might say, even if you put it somewhere that they sync and then sort of got to be there.
I mean, there may have to be different flows between different.
But one is the data, the other is the code. We have a question by Angela online. Have you found the need to revise previously existing annotations?
I think Jason will talk a bit more about this, but we have one of the major issues that we have is looking at the ensemble gene IDs and how gene symbols map onto ensemble gene IDs depending on what annotations are available. We haven't had the need to revise any as of yet. Like I said, this is still in the very preliminary stages, but I know that in the HCDCP, we have a lot of functions for updating ontologies, and we have done that there, and similarly in Cell by Gene. So if we do have to, there are pathways for it.
Thank you, Wayne.
Thank you.
Hi, everyone. Jason Hilton. I lead a project called Lattice. We do various HCA data coordination projects. Today, I'm in the capacity of, we're the lead curating team for the Cell by Gene data portal. Arthi gave a great overview, and so this is a great lead in for me here. And a first recognition of all the folks that this great big collaborative effort that is the Cell by Gene data portal. Next. And so one goal is to just share, visualize, download data. And the second goal is to enable the integration across these various collections, the different publications. And of course, that's going to require standardization. Next. And so I'll walk through some of the main standards that we have for Cell by Gene, and I'll call out some of the challenges along the way. This is our schema 2.0. But there's a few things in here that are kind of prospective that we anticipate adding for 3.0. So one thing, all data are available as both ANData and Seurat. Raw counts are required for all data so that integration is readily available, even without realignment. This is especially important for a lot of the data that have the raw sequence files and managed access. The feature metadata, Wade just touched on this. We require stable IDs. We use ensemble IDs, as these are the most common stable identifiers used in the reference annotations for single cell data. However, of course, everyone talks about, writes about genes as the symbols. So oftentimes we get a data set, we need to go from symbol to identifier, and we need to use the correct mapping. This requires details about the specific reference annotation that was used in the processing, and any manipulations to the gene symbols between the two time points. So that's a big challenge. Oh, next. There's our challenge. Okay, next. We have a handful of cell metadata that we require for every cell. You can see the required field. Most of them, we have an ontology that we require the values to be. Some of them have a subset of the ontology branch. Next. One big challenge, how to standardize ethnicity. That's hard. Next. This next challenge is about the cell type identifier. It's a lot of data sets we get don't have all of the cells annotated. Some of them, 10% were not the focus of the study, so they weren't annotated. There could be doublets that aren't annotated. Something like Visium, where actually each observation is multiple cells, and so that's hard to annotate. Or it could be a transition. There could be cells transitioning from one population to another. So kind of they're both, or they're neither, or they're in between. So that's a challenge. Next. Now we talked briefly. Melissa introduced this, like simplifying the ontologies. So now we've done all this hard work to annotate to ontologies. How can we make that tangible? So on the in-cut here is a new development stage filter we have. So you can group by adult. You can group all the terms that are under adult and just get that data. Next, and we're starting to look at tissue for this. So understanding how people want to search tissue. You could do it by cardiovascular system, reproductive system. You could do it by organ, like brain, heart, etc. So understanding what those higher level terms are and how to present those in the UX. And not just tissue, but it could be maybe we got it wrong on development stage, or it could be the assay. And so understanding how your users want to search and filter the data. And this is another way we use the ontologies. This is the SC expression that Arthi showed this morning. And you can see in the list of cell types, you get that hierarchical relationship. Next. So those are the main challenges we have. Thank you very much.
First of all, thank you, Jason. It's like we data, it was much easier than in
Thank you.
I think it was really smooth run. And the one question I have is actually the idea we had to use this data actually transfer to annotations like HCA arches or transfer labels between different studies, because so that we don't we don't need a consensus. We need different angles to approach a data because it's a multi-dimensional data.
Yeah.
Do we have any plans for that direction?
I know SC arches specifically has been discussed, but I'm not sure exactly what the direction is around that space.
Yeah, we should think on that. So we've been discussing obviously a lot how to things across whether you need another platform. We all have to sort of take a shot at the Azimuth, but they have some type of yeah, I usually like to have these actually at the users, you know. User interface, but sort of have a command line base. That's how we have it at the moment. So you can make it, you can transfer it. User interface is really that well.
So right now I have the idea on the microglial data. We have several studies differently labeled and different clusters, but I think all of them true. But actually transfer like these labels to the other study and see how this cluster nomenclature.
Yeah, I mean, I think there's a couple of things there. One is that you should probably keep all of these platforms need to keep the original text.
Yeah.
That the users added. Yeah, you shouldn't have it with ontologies. So the ontology might move towards consensus, but if the additional text often has information that we shouldn't be losing. So I think perhaps that's the compromise.
But I was thinking it's two-way. User can decide which annotation or labels on that data set from this study project is labeled on that data.
Yeah.
And so that you don't make the choice, user makes the choice.
Yeah, so we get the free text fields that David mentioned. And so I showed the minimum requirements. Users can add whatever other fields they want, and we make specific to preserve those free texts. So usually there's like an author cell type in addition to that, that preserves that. But yeah, you're right, the projection is a different
So
thing to tackle.
next up, we have Evan to talk actually.
There's a question online, how is integration performed with the raw counts?
So then each of those raw counts would be downloaded, and then you would, right now, the user does the merging. There is, so it's per data set download. There is a goal to work towards a cell database. That tile VB backend that was presented this morning is going to be key to where you can just download the cells as one data set, but right now it's individual data sets, extract the raw counts, and do the merge and
So there's no plan. I think it's very clear to do standardized automated integration somewhere.
correct. Yes.
And then second question online, if you provide joint visualizations, does new data get classified into an existing graph, or each new data set triggers to regenerate an enablement graph?
That is up to the user and the submitter. I'm sorry, the submitter. If they have an updated view, they can add a data set to their collection where here's version 2, or they can replace it. Right now, we give that flexibility to the submitter.
Yeah, another question with regards to ethnicity, do you think in the future this could rather be sourced from data somehow, some sort of genetic?
There is discussion about a lot of discussion that we have. The CZI has recently funded ancestry networks, and they're specifically looking at can ancestry, not ethnicity, but yeah. So I was talking about self-reported ethnicity. The ancestry networks are looking at, can we infer ancestry from the SC RNA and attack data? So yeah, it looks promising for that.
Next up, we've got Evan on the cat.
Yeah, and the other was discussing, so.
Yeah, so if you've got more questions, we can keep the discussion
Yeah,
again.
and somebody asked for the slide link, I'm sharing the screen, I can't post them, but if somebody else has the link, so please post. Evan, please.
Not very good. I was going to do a demo, but I think like David, I'm too used to being on Zoom, just sort of I pull up my demo. So I can just sort of review a couple of slides, as I think I had too many at that talk. I mean, you can look at the slides there, and I'll be putting up some YouTube videos in the next 72 hours ago or so about how to actually use this. So just based on some questions in the coffee break, I wanted to cover this, and I also want to cover some of the feedback that users are asking us to implement and sort of explain why some of this is a bit complicated. So most of the questions I had in the coffee talk were, people are very used to this idea about doing annotation transfer, things like custom and so forth. And I think these are great ideas, but really, to my mind, they're a bit of a hack, right? You're taking a selection bias. Well, you're taking a small reference that someone has told you is the correct reference. This is a high quality reference. And then you do some sort of k-means clustering to transfer those labels, right? So the problem behind CAP is actually, let's say we were to take all the molecular data we could possibly get our hands on, and then we were to push users to give us as much text as possible and related sort of metadata with these cell types. What could we learn from this, right? So you could actually learn possibly some distinct molecular signatures that are associated with those cell types and distinguish, let's say, cell types from cell states based on those molecular signatures. And in terms of the larger sort of community communication problems, if you push users to give as much metadata as possible, specific cell-related metadata, things like categories, synonyms, and so forth, you should be able to actually mine this data, right? So this solves the problem that there's oftentimes not a shared vocabulary for annotations. We're really discovering people who, when they upload annotations, there's maybe, they don't spell words correctly, or they use abbreviations and so forth. So if you actually have this sort of system, you could use basic text mining algorithms to learn things. So and yeah, I really want to, I mean, this is my community here, so I really want to push this on all of you and people who are watching online. I would like to know how to prioritize upcoming features. And what we have now is we have infrastructure working. This is sort of the bare minimum, and now we can start quickly adding features to make everyone here happy, right? So just to discuss why some of this is sort of complicated is it's very tricky at times to translate this into software, which makes sort of sense, right? So everyone usually tells me we would like some sort of evidence. When I look at someone's annotations, I'd like to see evidence. But thinking about this, what would that actually be? Do you want to see free text? Do you want to see, like, what would you actually do with this, right? And in terms of a UI, what that would mean is that for every cell that you specify with an annotation, we would force you to then write something out, like fill out a form, right? No one wants to use a UI like that. That's bureaucracy at its worst. It has to fill out several pages of forms for each cell, right? Things like community ratings as well. I think people like this idea, the sense of like, let's have the community rate what are the better annotations and the worst annotations. I have no idea how this is supposed to work, right? I mean, surely this is going to become somewhat of a popularity contest. You see that our favorite scientist uploads an annotation, this must be right, this is using my favorite algorithm, and then they put, thumbs up, this person I trust, maybe thumbs down. I'm sure you've seen Twitter recently, but scientists sometimes don't act that mature on this platform. So the idea that I would actually make like a comment section is something it makes me very worried. How much time do I have? Oh, 1 minute. Okay, we can keep going. Again, yeah, the evidence problem is something that I mentioned. Yeah, I mean, we just don't have. It's not clear to anyone what really is that supposed to be citations. It's supposed to be these are common markers I use. I really don't want to force people to give me markers because markers are they're not standardized even within humans, right? Between fields, certainly not standardized between species, I would say. I'm working on now like a UI for, let's say you take one person's annotation, you'd like to compare and contrast it to another. So I'd like to know what really are the features you're interested in. Would you like to see what are the different markers between the two annotations? Would you like to see what like different terms or whether they relate in some sort of way? I can build UIs to do this. We can do this fairly quickly, but I really need to know from you what should exist here. And yeah, just basic other UI requests as well. There's a question about people wanting to see UMaps, and I agree that the UMaps of cell by gene are very pretty, and I use them a lot. I mean, I don't mean that in a passionate way. Of course not. They're very impressive, but really you'd want to like do re-embeddings and so forth. That's usually what people want to do. They want to do reclustering and so forth. So building the UIs that can actually do this is our next steps. So better.
Thank you.
So exciting. So what I want to say, evidence. I think evidence, we need evidence. And the community ratings, it sounds fun, but it worries me a lot. It worries me a lot because you might have a lot of young people with very little experience. They might go and vote much more than someone with a lot of experience. So I'm a little bit worried about the community ratings. I mean, something to keep in mind. I think comments, as you said, would be really useful. So you understand people can't just, thumb up, thumb down. They need to explain. Evidence for me, the gene markers, I mean, there are some basics, at least some of the basics that no one should be debating. And if we start to debate that, then we're completely lost. And another thing that is in part related to this and what the others have presented is the difference between a cell type. Some of us define a cell type and a cell state as two different things. But I do think that there are some basic cell types that we do know exist, and we do know have certain markers. And that has to be, in my opinion, embedded into this.
Yeah.
And it has to be a minimum.
No, this is a good point. So I think there's 3 points there, right? So for the first thing, I think you're sort of singing to the choir. That's one of the reasons why I pushed back against the sort of community evidence thumb up, thumb down sort of thing, Twitter like, dislike, is that biology doesn't work this way. There are certain people who are experts in this field, and surely their opinion matters a bit more than someone who just logs in and starts, thumbs up, thumbs down. Would point fingers at young people, sort of throw stones, glass houses, right? But
No ages.
no, of course not. Right.
No.
So yeah, and in terms of markers, you're right, I think, but I think we should talk afterwards, because I'd like to know what is really the, I've been burnt by this several times, where I sort of think there are markers everyone agrees on. And I quickly realized this is not true. And
But then if you could just record them, then at least people could say, I called based on this marker. I mean, we've done that for jamborees in the past with the flight.
And that gets into a similar logic.
So some information is better than
precisely.
that.
Well, this is why we've been mining the molecular data as well.
That text mining through a lot of papers and see where somebody's going to use
Uh,
PGPRC immune cells and so on.
If you see how those algorithms work.
And I think,
But what I would do is what David was suggesting. So I have the molecular data, right? You know, we, the community, do obviously. So I would take that molecular data, I would be able to calculate those markers. If I see there's common markers coming up, that's what I'd be able to say that yes, these are. So I don't think we're ever going to be in a situation of either loss. Maybe we're lost now, but we're not going to be. I mean, there is the data will also be used, right? The third point you made,
I think we need to move on. So,
It's a really quick point. So you have a cell annotation like framework, right? That it looked like using a UMAP. Like, and so people are going to undergo certain steps in order to do the annotation, right? Like subsetting and whatever they're going to do, right? Maybe do markers. I mean, why don't you just track that? And have that be an editable, like, so people who like delete the stuff they don't care about, and then submit that as their like when you publish.
that's what I'm suggesting. It's like given that they give us the embedding and the cluster, we use that information.
Well, not just but the steps of it.
The steps, how would I record those steps? I would ask people to tell me how did they annotate this data set. So I use this tool, I spent this much time doing it.
No, but no,
if it's annotation on the tool, I think.
Right.
That's a really good point. We can put that down wherever there's annotation on tools, recording whatever can be recorded. So from the steps on that. Okay, I think we need to move on to Fabio.
I had just a short comment on Evan's thing. Can I do that, please?
Yeah, well,
Thanks. So if we want to build consensus, right, and we just have one data set from one lab, typically, and some applications, people kind of don't discuss it. But if you want to sort of put a full organ together, do we need to integrate the data first, or do we get people to re-annotate existing data sets? I think we have to think a little bit about that. So I think I could see CAP actually be used for the integrated one, where so then this consensus being built potentially more than for the individual data sets.
Yeah, I think both would probably happen simultaneously.
Maybe I both use space, right?
Yeah, I mean, this is something we've talked about with it as well. Sure. In terms of even what a priority. Right?
Yeah. I just tried. I would just think it's a good idea to have the option also to have integrated data sets in there.
No, this is our goal.
oh, sorry.
I think I should go first, right?
Oh, yeah, sorry, wrong order.
I can change slides.
All right, Angela next. Sorry.
Hello, everybody. Thank you, David. Can you advance the slides?
Yep.
So I'm just going to walk you through some of the tools that we have been working to try to automate the cell type annotation. Next slide, please. So I have been doing a lot of work on building cell atlas. If we can jump onto the next animation. And the workflow looks very much like we pre-process the data, we do the ambient RNA removal, we somehow get everything into consistent space, but then at this point we have an annotated data set. Next, please. And so what we have been doing is a combination of expert annotation, where we ask tissue experts to manually curate this for us. And then can we go to the next? But so then there is, when we are revising the atlas, some of the tissues have already been annotated. So we have developed a couple methods that can automate the process, but I think everything boils down to the next schema. Can we go next? Thank you. Which is, how do we go from these data set that we have some manually curation into the computer automated annotated data set? And a lot of us in the room have been working on methods, but I think one of the points that I want to make is that might be better to actually think about aggregating all the methods or some of the methods that have been published. And so what we have been working on more recently, if we can go to the next jump, is a tool called PopV. Neer's talk earlier today covered this a bit, and the idea with PopV is really to build a framework that we can create a score that help us understand what's the most likely annotation, taking into account all the manually curated data set that we have. So if we go to the next slide. One more. Sorry for the annotations, didn't realize how the format would work really well. So basically what we have in PopV so far is that we have 7 methods, which are the ones in the box that say popular vote. And we have an annotated reference data set in the case of what we have enabled for people to try out is with Tableau Sapiens. And so what we do is that we output a predicted annotation and a consensus score. Can we go to the next one? And where we take advantage of the cell ontology is on how we score the predictions. So we have co-developed one of the methods called onClass, and onClass is not the best method at predicting the correct label, but it's very good at predicting whereabout in the cell ontology the cell calling is. So when we have a tie between 2 methods, we add 1 point to the prediction that aligns with the on-class prediction. And I'm happy to go more in detail around here. And so with this method, if we jump on the next one, we have been able to, oh, sorry, can you go next? And next. So with this one, we have been able to improve the prediction scores across all the methods. Can we jump to the next? But I think one of the issues that we are going to run into very quickly is how do we take advantage of all the data that exists that is being added to the different repositories. And so what we need to work as a community, I think, as well, is to find an automatic way of translating labels into the cell ontology such that we can then run the algorithms and evaluate the predictions. If we can go on the next animation. And so with all this together, I think we will then have a framework that we can take advantage of the crowdsourced annotations from the different users. And then we can, sorry, there's some noise online. And so then we can use the crowdsource to update annotations based on consensus score and prediction. What we can take then advantage of having used the cell ontology as well, if we go on the next slide, is with these methods, we can, can we jump?
She doesn't need anything more.
Sorry, David, can you go on the next slide?
You can just click on it. I'm going to try to mute that one person.
And so basically, what we can do when we have the taken advantage of the cell ontology to help with our annotations is that we can then link the cell ontology tree with the molecular information and then computationally have predicted marker genes for all the cell types that exist on the cell ontology. And we have made these browsers available as part of the different portals that we have released with the Atlas. I'm happy to share the links. And then in collaboration with CZI on the last slide, something that we have been working more recently, there is a video, David, that comes up that you might have to press play. I think is working. So basically, somebody on the team that has now joined a CZI, Alec, it has added a couple of functionalities to a branch of a cell biology gene called XLX gene. And with this more interactive way of using cell by gene, we are hoping that we will have tools that the community will feel like using, and then we can harvest all the biological knowledge that I think is very essential to move forward on the annotations. Yeah, thank you.
Thank you, Angela. Any questions? Actually, I have a quick one, which is that the on class is good at finding where in the cell ontology term is, but not necessarily as good at pinpointing the precise term, I think you said. Do you think that's actually because most of the annotations we have available are actually to general classes, and if we have more specific annotations available, then the projection using the cell ontology would work better?
I think one of the issues with using the cell ontology has been the fact that yes, we have the general annotations, but also the lack of easy way of filtering cell ontology for tissue specific information. And I think if we could use the cell ontology at the tissue level and then train the algorithms on each of the tissues, I think we would have increased the accuracy.
That's actually entirely possible with a lot of the information that's already in there. I think we just need to make it easier for people to use. You have some APIs which allow that right now, which actually expose a lot more of that.
Hey, Angela, great seeing you. There has been this paper from Ahmed from Mahfouz's lab on tree classification. I thought that was quite a nifty thing and sort of allowed for more dynamic mapping and predictions. Have you guys been checking out that algorithm? I think might be useful for these sort of hierarchical things that we have in more and more.
I have not checked that paper, but thanks for the reference. I think, yeah, exactly. So those are adding those methods is exactly the point of building a framework like PopV, that we can use those to help improve the scoring.
I was thinking about that. I'm going to send that around.
Yeah.
Thank you. Yes, please.
So Fabian's up next.
Yeah, I just wanted to briefly show a few things that we are doing. Fortunately, I sort of need to leave this soon and for the stuff with Dana, there's no touring, but I really just want to be quick on that. So I think a little bit about how we use ontologies and how we sort of make them available. And for us, in particular, I think for method development has been very useful to essentially query and get a lot of data in quickly. And this is what we do with this Spire Toolbox, which essentially is a framework for quickly getting a bunch of data sets onto your end data and then quickly benchmark, compare the sort of load. It's not a database or anything, it's just queries from cell by gene or from BCP and so on. You just load different things in that allows you to very quickly manage these collections and then build this local data zoo as well as a model zoo, which we should have, where you can sort of have pre-trained models. And for us, a model obviously includes not only an embedding, which is kind of crucial, right? So I was asking Evan before, how do you put things together? Like the embedding is always integration, but then also the annotation. And the annotation can be manifold, could be also metadata. So this is, we have had a data syntax David in the audience who transitioned to independent postdoc soon at the broad has been pioneering and pushing this very much. This has been something super useful for us to essentially benchmark our methods, sort of upscale stuff and so on. And essentially sort of harmonized data management every lab is struggling with, right? Also you can sort of put in your private thing. Most of what we're talking is just once the project is done, but how do you manage all of these many private things that you don't sort of get to throw around? Has been for us an easy way. We can go to the next slide. Thanks. So this is the data loader that you can use for that. So essentially, literally, if you just want to get the live atlas in, we are all API based, right? So it's just a bunch of lines, and you have your data object integrated there for these things with the annotation. You can work with that. And what I think David we spent quite some time with is now to somehow sync with Evan CAP to make sure that we sort of could change more annotation, but more so also now to have this coupled to cell by gene to essentially have this transferred over and make sure that whatever we sort of wrangle in, I think that's how you call it, from other resources, that they're going to all be in cell by gene, so that somehow we don't need to sort of pull it together. Essentially here, a data loader is just some code where you get the data from, and eventually most of this could be, I think, in a very flexible subagent. I was hoping for DCP as well, but that's not as flexible at the moment, so that's why this is. But I think we need to have this code or this diagram of how we put things together and advise you. I think we should put something like that out and put on the website. So this is how we deal with this. I think just sort of for the people using that, I think on the database side, very useful. Maybe we could go next slide. So this is our thing, how we think there could be connections. So we have the collection server gene, you could get the data through there, and this is two different ways where things are stored. So also that avenue would be, in my opinion, quite useful to have this really put together. And I remember we were discussing direction of arrows. So I was thinking this is for me the most natural one. Let's just agree on something at some point and tell users, guys, this is how we think things together. And you can sort of highlight the important part and what sort of you can put your thing in the center. But I think have this to connect. As an outcast, obviously we want to do that. You know, this is what we actually there. The second thing I wanted to show, slide please, would be this. This is now something that I think, you know, what do we do with these annotations? Well, sure, we transfer them. You know, Angela, you've been nicely saying how you do this now for your cool big atlas, and he has been also demonstrating that. But I think, you know, we can only go so far. You know, in our head, we always need these bins. That's why we build this ontology. That's why we need to talk about. We need to have these beautiful labels. 100K labels, as you said, crazy. It's so big. But ideally, you know, the body doesn't care about. The body doesn't have big, doesn't have labels, right? So it's going to be always continuous things. So we need to have a database update of these things in some dynamic fashion. And this is a way now, not how we query cell types, but how we query on gene modules. You know, that's the typical orthogonal of transpose question you usually ask. What we've been asking now here is, can we come up with an architecture? Next slide, please, which is advanced, that essentially takes now prior knowledge and makes these sort of super flexible embedding models constrained to these priors. In this case, constrained to gene modules, but I think this could be quite related also to cell modules at some point, which is essentially what we do with the SCR-just transfer. Next slide, please. So we sort of take now, for example, some type of pathway database or also markers that you have for a particular cell type and so on, you know, these early gene cards ideas where you have this. Anything that you can give a name to, a human defined name, potentially also hierarchical, that we can then make interpretable. Next, please. So what we do, we regularize this matrix and make a constraint to this thing. Next slide, please. So essentially what we then generate is a latent space where each of these dimensions now have labels. And that's the key part. So you have now labels for each of these dimensions, and you can say, this is this gene program 1, this is gene program 2, and this is sort of how my UMAP looks like. And you can then the big one, and each of these axes has an ontology label. And that really helps people. And that's the first part. The second part, then I'm done.
Sorry, what kind of ontology label?
So in this case, at the moment, we just take, I don't know, a reactome or because this is gene-based at the top. But it could be also cell type labels. I think for one example, we just take B-cell markers or something like that. And this is Mo and Sergey is a bioarchive. You can check it out. I should stop now. It's the last thing I want to say. We can train, so we can select which ones are important for one particular thing. And we can also transfer this to a new data set and modify the gene programs or their ontology. So we can say in a data-driven fashion, because of this data set, we need to add this in that gene, or need to add this in that cell type. In this case, we update our programs. And this is sort of the data-driven type of thing. We have been super screening this, and maybe we just like skip those because I have one example and go over it. But I just want to say, so just skip that. So we have basically a tool how to then also extend this in a data-driven fashion. I think we're going to get one of these really biggest type of data sets that we have in bio. Like one we have now 30 million cells or something like that. Also trying. This is super useful. So we should update our categories with that. Obviously, those will be annotated. In this case, you can also detect new ones. How do you annotate that? Well, you should go back to CAP, for example, and sort of help us. So we need to render these things together, but I think there are some ideas how to make it. Yeah.
So I think it's one of the big challenges how we work between data-driven ontology development and also manual side. So we're doing a huge amount of data-driven development for the brain. We haven't been doing it outside of the brain so far. Too much to put into this session.
But data-driven is essentially clustering of
this is actually clustering driven and then projection, some of the projection from other modalities.
So I think in this case, this could be potentially time more flexible because you need to tell us if you want to represent the data, you need to add this gene, you need to have this cell type added. So I think this could go hand in hand. Maybe actually the brain would be a good example to try this out. For example, we did it only for review now, but I spoke to Ed yesterday. It's actually maybe a good idea.
Yeah, definitely. It'd be interesting to go with the chronological text.
You have this super strong ability, right?
Do you have a release cycle for atlases? Because oftentimes the atlas needs to go with the code, needs to go with the training that explains how this current conceptual understanding works.
Yeah. We are by construction media method group. So we basically update our methods and those we do, but whenever we have something new, we put it out. So the human lung cell atlas, which we've been now driving the integration, we've been having a sort of 1.0, 2.0, 3.0 plan for that. Timing wise, we did not really fix by something more, let's say, end of this year, we want to now add more data sets, but that's about it.
Yeah, because in HubMap we have estimates and then we have CAS tables and these to the reference organs. And so we now have a 6 months release cycle, but it's not always aligned with what many other groups are doing. And it's tricky then, and then some don't make it, and then they have to wait 6 months.
So I think,
so the HubMap lung is really not on that level, I would say, and it has been not as easy to
so I think would it be fair to say that the general problem we need to get down there is having version. So we've got all these different bits of infrastructure, all these different portals. If we don't have versioning on it, if we're moving between them and then annotations are shifting, then we're all going to be a mess. But if we can kind of come up with a consensus for how we version stamp this stuff.
I mean, we strongly believe in open access to many of these things. So, we put, for example, this app was also an Azimuth. So we sync with the role on that, right? So but we should put a stamp on it, right?
Updated.
Yeah, well, we exactly put it there, right? So, but then the curing tool from Raoul is a little bit different than this sort of more flexible deep learning one, but it's kind of okay still. So, the way how you use this is that determined by the thing. So that's why even though you might have the same version, the type of results you get might also depend on the algorithm that's been used. I guess you just need to be openly telling about it whenever you write the methods, then users can put that in.
I think it's sort of, and the last one is just a video, yeah.
Yeah, and I hope it runs. See, we didn't get to try this out, but we can try it. Yeah. So, Bruce, it's very crazy time right now.
Yeah, so Bruce couldn't make it.
So he could make it, but maybe if I could try. Yeah.
This is on, how to build infrastructure that uses ontologies and other types of integration to try and build atlases. I think it's quite complementary. Oh, is that good? We can't hear it now.
They should be able to hear fine.
We can't.
Yeah.
I don't. So maybe everybody has to watch it themselves. That's okay.
Yeah, I mean, maybe we're kind of really tight, so maybe we just need to move to discussion. Oh yeah,
So we actually have until 12:45.
12:45. Okay, so we've got 25 minutes, and I wanted at least half hour discussion, so that kind of works. So if you want to play around and see if we can find a way to get this running, then we can.
I think we can just have everybody.
Okay.
No.
So actually, if you want to move on to the discussion topics again.
That's a special guest.
This is how the discussion.
All right.
Yeah, if you want to forward the slide, I think there's a list of the. Right, actually, so I was a bit worried about trying to do 4 topics, given the time, and that's when I thought originally we could do separate breakout rooms. I guess there actually has been a little bit, I grayed out the first one because I thought there wasn't too much in the talks on this, but I think they have actually covered it a bit. So how can we extend and improve ontologies as our knowledge grows? So maybe we could do a short discussion on that right now. So I think we have a couple of things from Angela and Fabian that are potentially relevant to that. So maybe Angela, if you could comment on your ideas on that first, would you bring out stuff that you had from the talk, and then we'll open it up.
David, I couldn't quite hear the question. You want me to comment on number 2?
Oh no, if you comment on number 1, actually.
Number 1. So this is something that we spend a lot of time thinking, which is how far do we want to go in the tree, and keeping in mind the evidence. So there was somebody that I couldn't get, who was chatting about on Evan's talk about really the need for evidence when we are thinking about the cell type and the cell state. And I think that's something that we are also thinking very careful about if we want to extend the cell ontology. On our particular case, something that we have tried to do is to constrain ourselves of what already exists on the cell ontology. And then only if we cannot find, if we find that the existing cell types, the further we can go in the tree, doesn't satisfy or doesn't match the existing knowledge from the experts, then we are adding. And what I would very much like to see would be a way to flag gaps. So something that when we built on class, we were able to output that we haven't used at all, is a way of flagging that a prediction has equal probability between two branches. And so that probably indicates that there is some missing information. But this is basically where we are so far.
And so that maybe could point to issues with the cell ontology that could be modified because you're predicting something that falls between two branches. Maybe there's okay.
Yep.
Any more general? And then
super short comment. I think what we can do whenever we transfer labels, we need to quantify the uncertainty of the transfer. I think we have tools for that now. Multiple 3% in the KNN, but there's like a whole bunch of stuff on machinery that we can now be better at. And we saw, for example, if we transfer to diseases, the worst they get, like if it gets cancer, we're very unsure what's going on there. So this could sort of hint for, hey, there we need new annotation. Then you can really think if you want to transfer from some other, like organoid apples or so, there's something really fancy, or you can just get users to do the annotation. I think we should combine with these metrics to then have essentially some type of active learning that it's for the machine learning, so to have look into those areas where we do machine.
Question about so I think there's an interesting general question here. Is there an interest in, when we build atlases of visual atlases of brains or whatever, very often we nominate a standard brain. Here's a standard reference data that we're registering everything into. Is there interest for cell typing in sort of standard reference clusterings or standard standard reference among marker lists or some kind of standard reference? Albeit it's going to be tied to data, it's semi-arbitrary, but the advantage of having something like a standard atlas, like the brain atlas or virtual fly brain, is that everyone registers data to the same standard. And so at least you can say, according to our mappings into X, this is cell type Y. So some level of arbitrariness, I think, can be okay, given that gives us a common reference standard. So is there interest in that? I guess it's something we're doing.
Different people in different systems, that's why different opinions, but
yeah.
Adult brain, I'm very much in favor of trying to have a standard nomenclature, and it can be iterative, we can update it.
Yeah, I suppose it's for that standard nomenclature that we have at any one time, we point to version 1 of the standard mouse brain, and it was projection into that we use, or we point to version, the latest version of the motor cortex data.
I think there is, I'm sorry if I cut somebody out, it's hard to hear from the back. I think there is a strong motivation to have just that, to enable us to really start building on top of existing data sets. So something that I don't know how it's going to look like is the more data we add to the data collections that Sabajin, for example, is building. I think that once we come up with a way of integrating all that data, then it might be that we need to revise annotations, because once we might be adding resolution to the data in the way that right now we're doing annotations based on the neighborhood graph and out of the embedding. If we have more data, it's very likely that the clustering assignments will change. And so it would be really nice to have a way of tracking which cell labels are more reliable and don't tend to change so much versus other cell types, which we didn't have enough information, and those might be the reason why they are changing. And also, once we start adding and really improving the ontology itself, revising it, would be great to also automate that process. So let's say we find a new cell type that is upstream, for example, of some of the leaves. It would be good to have automated tooling that would take care of making all the existing references compatible with the new ontology just being released.
You can add to that. So I think, Angela, you really have a point, right? If we want to get a reference, as you also suggest, we need to have, I think, an integration. I think it cannot be just like a single data set, something like that. And of course, then, there's all these cross-tissue atlases already, so you kind of want to see how it maps into that. So we have these organ networks, the SPA networks this afternoon, right? I think one of the big aims we should discuss there, which ones are ready for producing such an integration. I think then that could be sort of turn into one of these potential standards where you then say, look, at least we map onto that with respect to that version, and maybe also with this release cycle. I think they can put them also professionally, but you should have that.
Yeah, I agree. That's really important. Actually, that's kind of what we've done with the brain, where the motor cortex mini atlas is actually an integration of multiple data sets in consensus across 11 different data sets.
I will say also, I mean, it's a function of how deeply you sample. So you do a shallow survey across the human body.
Well, if you just
Oh,
sorry.
Okay. I mean, it's a function of how deeply you sample that tissue as well. If you do a shallow survey of the human body, you haven't covered all the cell types by any means. If you do a super dense survey of the human brain, you pretty much covered it. You're going to identify leaf level changes, but not any major things.
So we sort of need deep coverage in order to, as well as consensus, in order to get a reference. Maybe we could move on to the next topic, which is how we make ontology annotation following standard schemas easier. So I think there was quite a lot of relevance there. I think there's an emerging consensus basic schema, which is the cell by gene schema, which is really a simplification, I think it'd be fair to say, of the DCP schema. And I can see basically we're using the same thing within CAP. So first question, I guess, do people feel that schema, as much as they're aware of it, is sufficiently expressive? Is this a good enough standard for us to be using moving forwards? Or
so I think the schema we see there is for data ingest, to keep good track of all the raw data that you might use or not use to construct an atlas, or for data that you want to map onto the atlas, but I don't think it's a standard that you would use to represent the atlas.
No, I think so that actually there's the ingest, and there's also annotation that comes with analysis. Which maybe is what you are more interested in there. I think so. There, mostly we talked about cell types. Is there anything else?
Oh, then follow up. So are you referencing like the versioning of the atlas itself?
Yeah,
So
but
your
also
atlas
different
product.
annotations. So you might have different tools which have all different cell type annotations. We'll need to keep track of what's behind each brain region, right? What data is behind there, and how was it done, right? And ultimately, you have to convert from a world which is tissue block, which is section centered or emotion centered, to a world where regions, some of the regions that are relevant for disease understanding.
Yeah, I think the versioning issue, something that's like, I mean, this comes up a lot internally in discussions with cell ID, and I'm sure it comes up a lot with others as well, but like, the question is, who's responsible for the versioning, right? It's like, it seems like that's, sorry. But I think that's a bigger question than cell ID, right? It's like, if we just say this is version 1, like whatever, like people will be like, who are you to say that that's version 1, right? Just because in your database, like.
Yeah. But I mean, I think so. Fabian's stuff on consensus, I think is really important here. So, you know,
but I'm
making a single data set standard is always going to be a dodgy thing. You've got 12 data sets in there, and these things are temporary. I mean, people can argue, you know, an oak building agreeing a standard parcellation scheme for a break. People argue to let work place. The fly one, which has been very successful, took 7 years. And we were promised it in the first 2 years, the first grant that we wrote, you know. So, but some, you know, the Alan has these parcellations. They're not completely consensus, but they're a reference. So now we can take all of the mouse data, we can project it onto that and say we can assign cell types to regions based on that reference. So I think having some reference with a version is better than none.
Right.
And you can always point to it. They can be different.
Yeah,
but
something like the genome references, where like the versions, and they keep improving them and they change the version number, but as long as you have something to track. But my bigger question is, who can do that, right? What is the entity that can come up with a tracking system and everyone can be like, okay, they're the responsible party.
I mean, it's a consortium that people are continually contributing to it. Maybe there at the back end, there's a firewall of some sort. There's a filter where it's like, okay, we've had so many contributions in the past months. Now we release.
Yeah, I mean, this is exactly what, you know, all of the databases do, Flybase or the MGI or any of the standard model organism databases, for example. You just have a freeze. You keep improving, you have a freeze, and then that gives you a reference. You know, so if you're doing an analysis with gene ontology annotations, you need to be able to say, I did this analysis against this version, because you will get a different result in years' time, which I think will have moved on. So I think just getting people used to having really simple, lightweight ways to make versions and making it really easy to put versions into their analysis.
Yeah, so kind of along that point, I think there's always going to be a need for like niche references. So think about lung right now. Someone could say, I only want healthy lung in my reference. Someone could say, COVID's kind of normal now, so healthy and COVID is my reference. Or someone could say, I'm studying long COVID, I want COVID as my reference. So maybe aligning not who is minting these versions, but guidelines for how to standardize and create a version so that you can reference it in your paper and have that provenance, right? That's what it's about, is provenance.
Yeah, exactly.
So maybe
I think that's, you know, so it doesn't mean that there's a single major consensus that everybody has to, but if there's one big one, one or two big ones, and then that regularly make standard releases, we can let many others bloom as well.
I would think that you would actually wanna before you go with the version of whether it would be like a healthy lung or the COVID lung, that you would want to spend some time harmonizing the two in the sense of finding which cell types the reference is very comparable. And so those ones could be, then we would start building this idea of reference cell types and disease cell types. And I think it kind of like comes back to this point of the need for integrating the different annotation references, because I think keeping each data set as a potential reference, I don't think it's gonna help moving forward the community, because more and more data is being generated. And I think what we need is this ability to classify into, for lack of a better term, a meta cell type. So we want to be able to classify an epithelial cell of the lung or endothelial cell of the liver. Would be nice to have done work on the integration before we start doing the cell type assignments.
Actually, I think there's general agreement on that as well. I think that was Fabian's point. So I think we need to move on a little bit. We're still sort of in point 2 a little bit. I think here on how can we make annotation easy. I think there's an interesting mix of how can we make a mix of prior knowledge from the ontologies, the context, and the machine learning, and sort of nudge people towards, because we throw whole massive ontologies at people, and it's too much. You're not going to get good annotation. So I wonder if there's any general thoughts on that.
So one question is regarding the integration of the current ontologies. For example, gene ontology already has a lot of annotation for cell types, KEGG, weak pathways, cell type DB, Pangal DB. All of those already have very well defined cell annotations. So how can we actually just incorporate that into ours?
That's a really good point, actually. So we actually do have, big kind of connected knowledge graphs of all these ontologies as well. So there's a knowledge graph which links cell types into the gene ontology, for example, and then we can link from gene ontology to reactome. So there's prior knowledge you can use there, but then there's also the annotations, the gene annotations connected with it. So that's really the gene set stuff that I think that that Fabian was referring to just there, where I think he uses something like that. So functional gene sets. Okay, so it's I think I think that's a really good point. Having an infrastructure which makes it easier for people to pull all this stuff together. And I think maybe that's where David's work comes in a bit as well. So David Fisher from Fabian's group is working on Aspera. So maybe do you want to talk a little bit about the possibilities for integration within that one?
So maybe for context, what we do is to incorporate these ontologies and these data or the scripts, which map unstructured data or any format data to streamline format flexibility. And we adopted 7 strategy and used everything essentially, and sort of increase the versions with big releases of our package. Maybe 4.3, like something I've been thinking. So if there's a situation where people are not happy with whatever the strategy or that specific version of the strategy offers, like I think that happened with NUM, for example, that the community was sort of like faster than the strategy. Something that could make this like a lot more structured and maybe also like more tangible for automatized applications would be to have like a standardized way of reporting cell types that are subtypes of stuff in ontology. So like thinking of something like, I don't know, like a Bobo file or something like that, that sort of like exists as an extension to the core ontology. But then, like somebody could write this and be like, yeah, this are like my whatever most fine-grained node of the ontology you're interested in, and just the 1 to 5 like this and this. I feel like that could be like as a type format, or that could be like relatively short. And then it's like really very tangible and then also
definitely could feed into, because we already kind of partly have that with we've got an extension called provisional cell ontology, which is basically a place for terms where we don't have enough evidence yet necessarily to make a full. So we usually like to have some kind of multimodal evidence. It's not just a clustering in single cell.
Is that for the entire community?
Yeah, I mean, right now we've used it almost entirely for brain. We've started to extend it a bit. I think there's a couple of different cells in there, but that's just starting. But actually, that's really the place for a lot of the automated cell type, new cell type calls as well as like a first stop. And I think then there's a big question over what the pipeline is to make sure to get stuff into the full. So they do subclass, they sit directly under the cell ontology and the same queries are available. They're just not as not necessarily as stable.
I don't want to change the subject, but we can sort of dance around this question for a while, I think. Has anyone ever tried to calculate how the current ontology is for data? So for instance, the GCP, we tried to map what a user has given us a cell type with cell ontology. And anything, let's say that when you upload, it's usually just like delete a cell and neurify, but very, very abstract. Right? So that's a way to start to measure how with the data we have, how
I guess the big question there is what counts as a cell type, and especially in the nervous system. So that, another big project of mine, which has been more consistently funded for the past 10 years, is the fly anatomy. And there we have 8,000 cell types and growing coming from data. But there we define the cell type by morphology connectivity, not transcriptomics. So I think you can go down probably quite a long way with this. So
the brain, but for any other organs as well.
Yeah,
So
I think there's going to be, Anyway, so I don't know how you'd actually. The question is, what's the baseline you're measuring that completeness against? I'm sorry.
So just another side comment that relates to all of the points in there is that we know that the cells, they cannot be binarized fully because they do differentiate so that there is the transition. And as we sequence deeper, we will also find new other cell populations. So one thing that I was thinking is that if you have an ontology, or at least a name for the cell that is actually based on the data rather than on knowledge. So for example, a T cell, if we can actually have an ID that matches to a 3D, 4D embedding of that cell in space, then you can actually, for example, whatever you call that cell always will have an ID, just like
and then I guess that's an ID for the instance of the cell. So here's my like this is an instance of a little thing, but there are many different things in this room. So I think we need to make that distinction. Plus, there has been quite a lot of work on trying to make these huge names which kind of embed many properties, and nobody ever wants to use them. So I think we need to distinguish between a programmatic artifact. A programmatic artifact, often one-dimensional strings are not the best way to encode them.
Yeah, but rather than grouping them by the general names, you group them by how they are in molecular in space.
Yeah,
so to create the hierarchy from the data.
I think that,
because then we can even add things. So for example, we have this cell, but now on the disease we have a B cell state. So everything can be captured.
The ontologies do allow for multiple types of classification to happen at once. So I've got a slide showing 8 different classifications of a single neuron.
And exactly. So then you replace that by the unique ID where that neuron belongs.
I think Ed's group is actually trying something to try it at one point and still running into computation. Right, so you do some sort of hierarchy and clustering. Every node you try to be like spit not suggest each one of those nodes I have actually skins.
So I think that we've hit time.
Yeah, that's good. That's a great one. Because in fact, the website's literature is so bad.
Exactly. Yeah, exactly. Especially working on trajectories, so you see like the names are meaningless. Like because you're really.
I agree that trajectories are a big problem in that. That was kind of in the first part of your question there. But how you divide that up? As soon as you've got a continuum, you have to make kind of semi-arbitrary choices about how to chunk.
Yeah.
And that does get difficult with any kind of categorical system, if you want.
I think having anchors is still very important. When you write a paper, a text, you need something short and informative. So everyone knows what you're talking about.
I mean, potentially.
Yeah, we had a lot of this discussion.
Potentially, you can combine sort of a very general term like neuroblast. If you have some staging system, you can make it relative to.
One question also for the group: who's working on knowledge graphs? Who's
was working on knowledge graphs on linked open data. So this is your way, right, to connect all these cool data sets. That was this book reference that you found in the beginning of the slide. So yes, I would love to talk a bit with some of you now. And Louis, really quick question?
Yeah, I just want to make a brief comment regarding this. So as an example with the average hand size, you know, we like love to relate with the average hand size, and turns out that no human on earth has the average hand size. So then we should maybe think of, you know, these kind of canonical signatures as exactly like there's a canonical, but it's like the no signature. So you can maybe like in the cell ontology, you can decide, well, you can classify cell. This cell has the canonical cell signature as presented here, or you can say this is a not all the so make a binary representation between the canonical signature and the part of the signature, and also question the signature itself and say, like these, like if you actually look at the whole data set of the cell atlas, you can see that cells of the canonical signature exist, and maybe the signature is also an archetype. Experts don't agree which ones are good markers. So I think it's we have the data to kind of like
sort of multiple things in metadata. We do actually have ways that allow to record multiple bags of markers for a cell type. And we're sort of moving very much towards that because there are multiple methods that can call out perfectly. In fact, there are probably very many ways that with good confidence identify the same cell type. So there isn't the perfect set of markers like that. But also the ontology allows for there's much variation on that. It just gives some level of constraint and says with some reasonably high level of confidence, these properties will apply. It doesn't, it doesn't prevent cells or whatever, all sorts of variations being classified under that. So there's that flexibility in there as well. Yeah, sorry, we're about 5 minutes over time, so I think so another.
I just wanted to do a lunch call because we have a cell atlas meeting at 1 for those in that session. The Get Atlas networking session is exhausting.
Thank you all. There will be morning. I think some of us are planning to be at 8 or earlier than normal, and it's the hour and balcony. So if you want to join that, please do come. And I guess there's some overlap.